It's really great to be here. Let's see if we can get this to work a little bit better. And really an honor to be following Stanley. A little bit intimidating, but it's really nice to be able to give my perspective on really where I see the future of cryobiology going, and sort of share in some thoughts along this topic. And to get started here, let's see. No. We're in a little technical difficulty. Okay, so this is Janus. Janus is the two-faced Roman god that could see both the past and the future at the same time. I think that Janus is a very appropriate mascot for not just this presentation, but also for cryobiologists, for two reasons. One, I think we as cryobiologists are I think, very good at looking to our past and really building on it. And also, this was the Roman god that could step in and out of time. But if you think about it, this is what we do as cryobiologists. We freeze things and suspend them, and then we're able to reanimate them later on and really step in and out of time. So I think it's appropriate, even though I'm talking about the future today, not to re-describe what Stanley just talked about, but to really, by way of segue, talk a little bit about our past and where we came from. And I do like to sort of point out that I really think Hippocrates might have been the first real cryobiologist. If you go back to some of those early writings, he was really the first fellow attributed to describing using temperature to modify biological systems, specifically stopping swelling and sort of slowing down bleeding. So he's really the first guy to kind of link biology and temperature together. Now, quite a bit later, we start getting into the stuff that Stanley described, and things like Boyle, when he described the fish and frogs that could actually live with part of their bodies frozen. Then you had quite a gap, really, in study of cryobiology. But most of these early efforts, I wouldn't even say were empirical. They were really observational. And that's how we got our start, was really in an applied kind of way, looking at what happens in nature and sort of observing it, and then trying to apply it like Hippocrates did, if possible. Now, if you sort of move to the modern times and really get into the times when Luyet started to describe vitrification, again, there was this big gap where I think there were, between those early writings by Boyle and probably the early writings, and this was referring to when he published his book. Obviously, the journal came out a couple years earlier. But in between those time periods, there was only about 100 papers, I think, published on the topic of cryobiology in those several hundred years. So quite a gap in the literature, especially if you look at the logarithmic escalation that Stanley showed in his talk with respect to today's modern time and uses of cryobiology. But obviously, since that time, we had the discovery of the cryoprotectants, and then really, I think, another key contribution was when Peter Mazer really started to talk about what we now call biophysics, I think, in the discipline of cryobiology, really using a lot of the observed quantifiable variables to go on and do quite a bit of work to improve some of these systems. And this has gone on to be really worked out pretty well for a lot of people, including myself and others that have used some of those early sort of underpinnings to model systems and figure out how to make protocols better and better. So what would Janus say now about where we're at? Well, we do nowadays have computing power on a scale that was never conceived of when these early cryobiologists began doing their work, which does make things like modeling easier to do. We also have some very sophisticated technologies for observation, starting back with Ken Diller and cryomicroscopy, and then going on with things like Jens Carlsen's unbelievably fast frame rate analysis to outpace sort of natural phenomenon that we couldn't see before as systems freeze. But in general, even with all of this, as we continue on, we see the iterations really just become less and less significant, I would say. So just doing the same thing over and over again has really led to sort of a refractory scenario To some extent, this is in part if you look at how we perform science and how we should perform science. Really, we're trained to look at mechanisms first and then applications second. This is to obviously come up with a logical hypothesis that we can test and then to apply that knowledge. So we're doing things correctly by following the paradigm we're following. However, it's never been that straightforward in cryobiology, as we all know. So I'm not advocating at all breaking the scientific method. I'm just indicating that it's been a very complex development in our field. And I think that's really given because of the fact that cryobiology itself is so complex. And really, this was said best, I think, by Harry Merriman in a recent book. I think this might have been one of his last publications. And in fact, if you're a cryobiologist and don't have this book, you probably should get it. It's actually a very, very good book. But Dr. Merriman in this passage here describes that really to understand what's going on, we're going to have to account for interactions down to a molecular and I think probably an atomic level. These are things we really still can't do. Even with all of our technology, we're still not at this point where we can do this. So that being said, today we still do quite a good job at empirical and theoretical approaches, but over time, more and more natural cryobiologists have been publishing and learning things, and really it's now to a point where I think these are truly ready to start beginning to intersect, more so than ever before. You've had natural cryobiologists, you've had the medical-oriented specialists, you've had the agricultural people. I think we really are at a time when all these things are really on the verge of converging. I think we have to be careful to remember that what we've been doing is really manipulating the things that are convenient to us. Most protocol development over the last few years, at least, and I'm guilty of this, it's focused on really capitalizing on what we know about the colligative effects of cryoprotectants, dehydration, standard approaches to do things like make it easier to add cryoprotectants and remove them, looking at different cooling rates, inducing ice nucleation. We haven't spent as much time on the applied side looking at ways of biologically suppressing metabolism, looking at ways of inducing stress responses. I think there's a whole series of work on this that has not had as much influence on the clinical side of cryobiology as it perhaps should now. So I think with that said, it's time probably to get a little bit more back to nature. So if you continue on with Dr. Merriman's quote, really, natural selection has been an extreme example of empirical research. It's got a head start on us that we'll never be able to catch up to. And really, I think he said it best. I won't read the entire quote, but again, the number of trials and errors is astounding. But what's really good to look at is the fact that this might not only reveal the why, the how, but also the why. So in essence, we can skip a lot of that basic mechanistic study and perhaps borrow from nature and start way ahead of the game if we do it appropriately. So what does Janice say about where we're going? Well, I think it's easiest to divide this into two real kind of broad topics, and this will make up the rest of our discussion today. Really, what will drive cryobiology in the future, and then how will it work? So if we talk about what's going to drive this, and I'm not advocating anything, I'm just sort of calling it like I see it, basically, in terms of where funding will be, both from an academic perspective and from a corporate industrial perspective, and what really is kind of emerging as very important problems where cryobiology is very, very relevant. And I think you can summarize these by these three broad topics. You've got cryomedicine and cryosurgery, you've got biobanking, and you've got cell therapy. And you still have all the basic research, but the ways that these discoveries will find meaning in the applied world will probably be in some form of these categories. By organ preservation or cryomedicine or cryosurgery, I'm really talking about organ preservation, so hypothermic preservation of organs or perhaps organ banking, as well as its counterpart, which is really cryosurgery, or using what we know about ice to destroy tissues. Incidentally, we have two sessions at this meeting that I'll plug, Session 2 and Session 15, that will talk about these very topics, and we have a very special relationship with the American College of Cryosurgery with John Boust here, and you can see their website, but It's an area of cryobiology that we're really kind of starting to see emerge again, and will be very important in the future. Now, when we talk about biobanking, we're really talking about repositories for preserved specimens and data. And this is literally exploding now, where you have research, pharmaceutical, conservation efforts, all banking as many tissues and samples and specimens as you could imagine. Now, what they're only now starting to realize, I think, is that if you put garbage in, you get garbage out. So we as cryobiologists are becoming very, very relevant in this space again. Even proteins that were thought to be very stable, we're learning are much more labile than we ever might have thought. Genomic material, much more susceptible to damage than we ever thought. We have a couple sessions at this meeting on this topic as well, sessions 11 and 16. As Eric James mentioned, these are in conjunction with the International Society for Biological and Environmental Repositories, and you can see their website there. But it's an area where cryobiologists will become very important in the years to come. Also, cell therapy, or using cells as pharmaceuticals. This is also literally exploding in global markets and in mainstream clinical medicine. This is behind biorepositories because there's a significant regulatory burden here that's not as relevant on the biorepository side. But it will happen. It will be part of mainstream clinical medicine, and it will require cryobiologists. There are groups, the International Society for Cell Therapy, also working with us in this meeting, and we have a session tomorrow that's all day, actually talking about how to apply cryobiology in these settings. I think what a lot of people sort of realized was they did all this work figuring out how to get cells to do what they wanted them to do, how to use cells as drugs, how to make them in massive scale, and then forgot to think about how to package them and freeze them and distribute them in a frozen state. And we're finding that now as cryobiologists, we're extremely relevant to this. How will this work? How are we going to get there? I think we go back to what's always worked historically for us. Look at the Janus face backwards again. We do basic research. We continue borrowing from nature. We apply all these tremendous new technologies we have. We really delve into what we're learning now about the physical sciences as well as the biology side, and go back to our true interdisciplinary roots. I think cryobiologists were more interdisciplinary than anybody. They were doing those types of cross studies before there was a term interdisciplinary science, really, with the engineering and the physicists and the chemists and the biologists. So I think as we do this, we have to still focus on the biophysics, we have to still focus on the things that we know we can evaluate, but we also really have to, again, bring in the natural world and borrow from that. But we have to remember that evolution has basically been blind, and it does have that advantage of billions of years. So it's really looking at, you're talking about huge multi-gene combinations that are selected against or for. While what we typically are used to doing is maintaining a single variable and modifying others. So we can't just really count on lifting exactly out of nature and applying it to our systems. That's just not going to work. If we can get guidance or use the spirit of how nature really does this, I think that's the better route to go. Now, Professor Story has, over the years, in elegant ways, described a lot of natural systems. One of the most familiar ones that I'm most familiar with are the wood frogs that can survive some form of freezing. And really, you can sum up a lot of that data into really three categories of how they've adapted to survive sort of a natural cryopreservation process. These would be cryoprotection, but then suppression of metabolism and stress response. All of these things happen naturally. The frog does not expose itself to 10% DMSO. It produces glycerol and other sugars and things like that metabolically. Meanwhile, other genes are activated that suppress metabolism, that allow it to prepare for these great stresses. And, I think we're starting to see naturally this convergence. There's some other really smart people in our society. One reference here, Mike Zieger, who's actually just down the street from me in Indianapolis, who's now been working with looking at human endothelial cells and really trying to elicit these same kind of responses in the endothelial cells. And what he's shown, and this is open source paper, so I'd encourage you guys to look at it if you're interested. And like most of the work that Mike Ziegler does, it's very, very thorough. But what he's really been looking at is by very simple treatments, by just really exposing cells to cold temperatures, not even very cold, really more like ambient temperatures, for 72 hours, he really modifies the actual phenotype of those cells. They respond completely differently. They produce new proteins. They behave differently. And it really boils down to these two mechanisms which address in one fell swoop two of those three adaptive responses that the stories have described, namely suppression of metabolism and induction of stress response in a positive way. This is amazing. He's putting them in a refrigerator for 72 hours and then they suddenly work. Why are we not doing this? These are the kinds of things we need to begin to really start to look at. These mechanisms are just there for illustration, but what this is really starting to point to is another kind of a cool situation where if you could take this and combine it with work of other smart people in our society, like what Jason Acker's done with Mehmet Toner and Ali and others, looking at different ways to sort of either genetically induce or permeabilize cells to accept natural cryoprotectants like sugars, and then you really do have that sort of natural system reproduced in a clinical environment. You now potentially could have cells that you could cryopreserve with minimal manipulation, thaw, and transfuse into a patient without doing any post-thaw processing. That's really where we need to go. And this sort of evolving version of looking at cryobiology, I saw this and really liked this term, Keith Harding, cryobionomics. So sort of jumping on the bandwagon of genomics and proteomics, but really looking at the stability of the genome over the course of cryoprocessing, storage, and recovery. With all the work we're doing now, understanding epigenetics, we really need to pay attention to this on the cryobiology side and start to really exploit this to our advantage, if at all possible. And this goes to some pretty powerful points. Just last year, the Nobel Prize was just won by the combination of these two fellows here, Professor Yamanaka and Professor Gurdon, and really what they were looking at is the same kind of thing. They were looking at it in extreme form, but based on Professor Gurdon's understanding early on of molecular biology, it really made it possible for Professor Yamanaka to induce cells to become embryonic. And that's a tremendous thing. It's a tremendous scientific accomplishment. What we're trying to do is pretty difficult. I don't think anybody in this room would want to go back to work after being exposed to minus 196 degree temperatures, and that's what we're asking cells to do every time we freeze them. But it is much less difficult, I think, than what these guys did. I think we really can probably look to molecular methods to, instead of modifying protocols, especially as we're getting to a point where it's becoming more and more refractory, we can modify the cells to fit the protocols. I think that's really a direction we should, again, try to consider. And we also need to keep looking back again to our interdisciplinary sort of roots here. And I just want to kind of highlight a couple things from this standpoint that we've been working on. It's a combination of an Asian group and a Western group. I'm not sure if we're going to win the Nobel Prize for this as the last group did that I just mentioned, but still, it's a pretty cool set of experiments, I think. Working with Professor Sumida in the Sumida Clinic, Seijo Sumida, who's here at the meeting, we wanted to look at how we might be able to really try to start to bring cells out of cryo that have been suspended for a very long time and maybe potentially damaged. So every year we'd heard Professor Sumida at these meetings talk about bone marrow or blood that he had frozen for just decades. He was one of the pioneers of bone marrow transplantation in Asia, and he had many of these samples frozen down that were subsequently donated. So we actually went in and talked to him about this, and these were all frozen for basically hematopoietic cell reconstitution. So if you need a bone marrow transplant, typically what they're looking at are the hematopoietic or bone and immune forming cells. And all those early protocols and the work that I focused on as a postdoc were really focused on these cells, on the what they term HSCs. So back when he was freezing these samples, they didn't know really about any other cell type in there other than the other regular blood cells. They didn't know about any other real stem cells. And then over time, people really just determined that there was a whole entire category that they were missing of cells in this bone marrow called mesenchymal cells. So he was freezing down things that he didn't know he was freezing down using protocols that were not developed for these cell types. So we wanted to look at possible ways to still be able to capitalize on those cells. To do that, he agreed to send us some samples. So again, this is a collaboration among cryobiology investigators who only know each other from this society that are in completely different generations in completely different parts of the world. But we were able to work together to get Japan to allow us to do this, to get the United States to allow us to do this. And he sent us samples of bone marrow that was cryopreserved 30 and 40 years ago in dry shippers from Tokyo to Indianapolis. Now, as always, there's going to be problems. The very oldest of the samples had broken. He repackaged them as best he could. These did initially look promising. They did seem to begin to show some development. However, they did not take off, I think, because of the repackaging. But the 30-year-old specimen did. And these cells went on, and we didn't just look at this to see whether or not we could get cells to grow and say, good job. We wanted to see if you could actually scale these up in ways where they could be applicable to true industrial type cell culture work. Really looking at some of the ultimate stability studies. To my knowledge, nobody's done anything at this length of time. This is not a tremendous scientific achievement. This is just something to demonstrate the real durability of cryopreserved specimens. And as we talk, if you come to the sessions tomorrow on the GMP side, you'll get a sense that a lot of people outside of our society do not understand what we do. And it's one thing if you're an academic investigator and people don't understand what you do, and you can say, well, at least the people reviewing my papers do, and hopefully the people that review my grants do. It's a whole other issue if the FDA doesn't understand what you do, or the CLIA inspectors, or whatever regulatory body makes your life difficult multiple times a year, like they do for ours. And when we have people from those different agencies come through our lab and ask us about cryostability, this is something that normally is just sort of glazed over and glanced over in their other inspections, and it's obviously an area where I like to sit down and just start talking about it and tell them about how cool this stuff is and how great all these technologies are and how good our history is. As soon as I tell them things like, well, there have been very good papers with accelerated aging by bombarding frozen embryos with radiation, and these match up to theory, and I pull out the paper that Peter Mazur hand-signed to me, which is the first record that I've seen of some of these things really kind of spelled out in a way that makes sense, they don't believe anything I say after that. I tell them that these samples are likely good for hundreds, if not thousands of years. Sure, we don't have stability data that long, but we can't. We haven't been around that long. This is the first time that I know of where something's been frozen for this long that can be expanded in ways clinically applicable to really get those guys to just leave us alone, to get off our backs. So, I think it's a really good way to, again, try to use practical approaches to get where we need to go. Now, as these cells developed, they grew very similar to the cells that we're used to looking at. Now, these cells were taken from very ill patients, so there's going to be some potential issues there. But again, not just thawing out cells and seeing if they're still alive, looking at trypan blue. We took these and grew them out, and they're growing very well still. I think we have one of the guys from my lab here who did this work we're doing the flow cytometry and now differentiation of these into bone, fat, and cartilage. And we're now, while I'm here, reaching out to the NIH to see if we can get some support from them, actually just collaborative support to do molecular characterization of this and really do show that you are able to achieve very long-term stability with cryopreservation. Now, really to spend a little bit of time on sort of how to conclude here, I do think that the future of cryobiology is very, very bright. We do need to think about biological directions that include these molecular approaches that really tie into how we can naturally do things like create osmotic tolerance and develop stress response and really protect mitochondria, protect the avoid reactive oxygen species, all of those things. Something that I didn't spend a lot of time on, and I won't today, but equally important are these physical directions. Vitrification is really something that you go to meetings. Again, as a cryobiologist, we go to these meetings outside of our discipline, and often, I think if you're like me, you get kind of frustrated. The big reproductive society meetings that there are in the United States, I think they started talking about vitrification without really knowing what they were really getting into. And a lot of these people that are describing vitrification procedures, I don't think that's really what what's happening, or that's not complete vitrification, or it's just odd terminology. But we really need to spend some time on this. We have so much technical power now. We probably can begin to start looking at questions that were kind of put aside decades ago because we just didn't think we could achieve that. Things like temperature rates, cooling rates, warming rates, things that we used to say, well, you could vitrify water, but nobody could ever cool things fast enough. Well, maybe we can. There's some really great topics, some great papers later today even talking about nanoparticles and microwaves and ways to really do things that we haven't done in the past because we didn't have the tools. There's some real pioneering work being done in vitrification. You heard Professor Kuleshova earlier. She's done a lot of this work. A lot of people in our society have done this work. We need to continue to not just branch out into areas that we're familiar with, but branch out to other groups that we're less familiar with, I think, as well, to continue our future down this path. And in general, as we look towards nature, I think we should also continue to work together. I think, again, one of the real strengths of our society has been this interdisciplinary nature and the fact that we have a meeting with such diverse groups of people. I really have not been to anything else like this, where in one session you have somebody talking about fish, egg cryopreservation and coral conservation, and then in another meeting you have somebody talking about cryo-obliteration of prostate cancer. I mean, this is really a tremendous situation we're in that we need to capitalize on and really need to work together. And that's really the end of my talk. I'd be happy to answer any questions for you guys. I wanted to make sure that we gave Stanley plenty of time for his first one, so I think we might be a little bit early.
That was excellent, Eric. Just a couple of points. If you look at what we have successfully frozen and what we have not successfully frozen, the non-successful ones are pretty dramatic in a sense. We have frozen mammalian systems pretty successfully, and that there's a great paradox because the mammalian systems are the most homeostatically bound of any of them. They require very specific temperatures and pH and so forth, and yet those are the ones we subject to these extreme non-normal situations, and they survive. The ones we have not cryopreserved are the non-mammalian vertebrates and the entire group of invertebrates, plant mixture, some yes, some no, algae, a few yes, a lot no. And so I think one of the things for the future is to understand why it is of these great problems in freezing the non-mammalian systems. And that's, of course, we have really not been successful at all, or hardly at all, in even mammalian systems, freezing complex tissues and organs so that the intercellular systems have a degree of complexity beyond that of single cells, and we have not been successful in understanding that. So I think that those are areas where there's tremendous importance and tremendous potential.
Yeah. That's a very, very good point, especially when you start talking about conservation and those areas. And now this is kind of wandering outside of my area of expertise, but just a couple of thoughts on that. I think it is sort of odd or ironic almost if you think about the fact that you have something like for instance, fish species where their eggs can be released into salt water, yet we can't figure out how to manipulate them in ways that don't destroy them during the addition and removal process with the cryoprotectants and those kinds of issues. And the same with plants that can survive, maybe overwintering, but can't survive cryopreservation as well. I think some of that goes. But the only thing I can kind of comment directly on is to say that I think that point that I made about not trying to, not expecting that we can just lift some of those adaptations and apply them in areas directly to cryopreservation. There's so much going on that we'd be blind to do that. So, for instance, with things like plants that have cell walls and they seem very sturdy, as they cool down, I think their membranes kind of reorganize themselves to survive that better. But that lipid change actually makes them less able to survive actual freezing. So those kind of situations we've got to somehow address, and it actually, what seems like it should be easy, actually becomes very, very difficult. So that is going to be a big part of the future, I think.
So Erik, is it on?
Sure.
All right. So Erik, the demand for our discipline is ever increasing. We see it in the medical side, we see it on the agricultural and conservation side. But from a basic science perspective, are we keeping up with the demand? Are we moving fast enough to adopt new technologies? And if not, how do we as a society reposition ourselves to be more relevant for these groups?
That's a good question and an interesting way to put it, I think. I think that we actually are keeping up with the demand scientifically and less so in an applied way. I think there's stuff that we're doing that is really tremendous that ought to be used. And I think we, by forging some of the relationships, and even at this meeting with the ISCT and ISPR, I think we're trying to do that. I think we're addressing that, but that will be a huge part of the future, is making sure that we keep people aware of what we're doing. I know you and I both get frustrated at places like the American Association of Blood Banks, where they talk about problems that we've talked about before you and I were in the society. They were talking about. So that's a very, very relevant point.
I'm a clinician I'm not a cryobiologist, and I'm not versed in, I don't have the ability to do the kind of research you're doing, but I'm applying it all the time, every single day. And I can see huge benefits to it, whether it's ovary freezing or egg freezing for our infertility work. But also, we found many labs do a very poor job of freezing their embryos, and that they get sent to us. And and I found Dr. Mazur's and Lebo's points are very valid. If we just really do a super rapid thaw, no matter how poorly they were frozen, we're really getting good embryos out of it. And so there's a lot of practical applications. I don't have to necessarily understand the complexity of the physics that you guys have worked out, but I see huge applications because you seem to be simplifying for us what we have to do, even if we don't understand how you came to that.
And I think that's a good point. I think that is to some extent by design. I know we have a sperm bank, and when we were very small, on weekends, I would deliver the frozen semen to the clinics with our IFU, our instructions for use, and I'd sit there and watch them then not look at it and do whatever they do, and usually I would say, no, that's not how you're supposed to. So you have to spend time making sure. I mean, as cryobiologists, we're always worried about freezing things. Everybody that uses our technology, well, usually their first question is, how do you thaw it? It's easy to freeze something. How do you get it back? Is the way that it's looked at. But if you do put the work in up front, and it's incumbent on us to make robust systems that are able to achieve the same results almost in any way of applying them. But there's still a lot of work to do, as I'm sure you see every day.
Oh, yeah, for sure. Thanks.
Erik, the point you made about, about time frozen, I think is an important one, and one that perhaps we need to pay, sorry, break the machine. We need to, perhaps the members of the society could begin to tabulate such results. Now, there are a few people who are doing this. Chris Walter in Fort Collins is doing this with plant cells, but I have to relate a little anecdote. a couple of years ago, I was at a meeting of the American Society for Reproductive Medicine talking with a physician, and I told her that we had thawed some bull sperm that had been frozen 37 years earlier. We could document when it was frozen, how it was frozen, the storage time, all of those things had been documented. And I said, and we demonstrated that that sperm was functional, it would fertilize eggs, and the eggs developed in vitro into blastocysts. And she said, oh, but that's animal sperm.
Yeah.
That doesn't have anything to do with human sperm. Now, of course, there are data reporting the birth of a child from sperm frozen, as far as I know, the longest time, 28 years. Yeah. There are, the people at Jackson Laboratory are beginning to thaw mouse embryos that they can document were frozen in the middle 1970s. But it seems to me that these, time is passing, and these data ought to be tabulated to demonstrate, in fact, that the concept that a sample once stored can be held, quote, forever.
Yeah.
At the first embryo meeting that was held at Jackson Laboratory in 1974, Peter presented a point on this item, and a radiation biologist, Russell Dufresne, talked about the fact, based on radiation effects, he would estimate that the storage time for an embryo was a minimum of 3,000 years. 3,000 years would be long enough for cosmic radiation to cause double-stranded breaks in the DNA.
Yeah, that's a really good point, and it is what I think is so amazing about our group, and it also, my former advisor, who's since passed away, John Kreitzer, he would always remind me to remember that, because that means you're also preserving your mistakes. Whatever you put down is exactly what you're going to get back if you did it right. So if you did a bad isolation or something, you need to worry about that too. But I think you're absolutely right, and I think we've been discussing, some very motivated people within the society have been discussing trying to do some position papers or some kind of papers that describe some of these things to really do that. And perhaps working with groups like ISBR, we could get more help. I mean, it's really an effort that's going to be fairly tremendous. But we've had enough history now that there are so many of these that it's worth doing.
Dr. Woods, thank you for a very informative presentation. My name is Lou Jones. I'm an electronic engineer and a mechanical engineer, and I got into this field quite by accident in 1985 on the basis of one phone call from UC Davis regarding an embryo freezing machine, which had malfunctioned. I think that the stressing of the cross-pollination of different technologies is really helpful in underscoring the need for that sort of thinking to be a continual process in the evolution of our discipline. For example, in the 80s, I was on a plane trip, and I thought about the wire labeling systems at use on altitude as surviving minus 60 C, and also the need for reliably labeling straws at cryogenic temperatures. And I thought, well, if they survived at minus 60, maybe they'll survive at minus 186, and lo and behold, they did. So we brought a product from a completely different industry into the realm of cryobiology that really filled a need. And I think that sort of thinking is really key in moving forwards in the future. Thanks.
That's really, really true. And I'll point out one area I didn't talk about, but another cross-discipline is food. There's been a lot of work done to preserve food, obviously, and you're in essence cryopreserving food to some extent. I mean, they call it I can't remember what the exact term is, but basically when cells lyse, they lose flavor. The chemicals are what give, the intercellular chemicals are really, and the proteins are what gives the food its taste. So there's a lot of work being done there, and we're looking on an industrial scale as we're talking about freezing trillions of cells at a time. If we're getting to a point where we're doing huge scale production, you can't use a planer. You can't use a standard cryomed. You need to use high throughput devices, and the closest we can find are how they freeze turkeys. They put turkeys through big laminar flow nitrogen freezers, and they cool them down pretty quick. So all those, we need to keep our eyes open. You're exactly right.
Eric, I really liked your concept about biobanking. I mean, being a biobanker myself, there's so much that we need you folks to help us with. We're trying to come up with best practices, but our best practices in the biobank are all anecdotal. You folks have the science. So one of the goals that we have to be here is really to establish collaborations with you folks. Peripheral blood mononuclear cells, that we'll talk about later, I think on Tuesday, being able to come up with better cryo cocktails and better freezing and thawing protocols and rate control freezing protocols that we've been using for years, but we've never really looked at the efficiency of some of these and success of these. And we haven't been customizing things. And we're finding that some of our cells aren't surviving as well out of the freezer, and of course, as we ship things and manage them, these are all very application-driven, as you're mentioning, that we need help with. And in order to really establish best practices, we need the science. So we're hoping that at the end of this, there'll be a lot of collaborations that will come out.
I think there will be. I think there will be.